Melanoma is one of the deadliest forms of skin cancer, and UVA radiation from the sun have been implicated as a major environmental factor in its pathogenesis. Categorized mainly to three types, UVA accounts for 95% of the UV radiation that enters into the Earth's atmosphere, penetrating deep into the dermis where melanocytes resides. Prolonged exposure to UVA radiation can stimulate excess production of reactive oxygen species and lead to potential malignant transformation of melanocytes. Hi, my name is Victor Rowland, and my research was on investigating the antioxidant effects of ellagic acid in mitigating UVA-induced oxidative stress in melanoma cells. Ellagic acid is a polyphenolic compound found in pomegranates and various berries, and research have shown that it has promising therapeutic ability in mitigating UV-induced damage in skin cells. Here it was hypothesized that EA will reduce UVA-induced cellular damage by decreasing RS production and DNA damage. The UV photoprotective property was evaluated using UV spectroscopy, and the results showed that EA absorbs strongly within the UV region, with maximum absorption peak identified at 280 nanometer. A375 melanoma cells were used as an in vitro cell line in this study. Ellagic acid was administered at 3 to 40 microgram per ml concentrations, and after 24 hours of incubation, the MTT assay was carried out. The results showed that ellagic acid had no statistical difference compared to the untreated control. Hence, the 40 microgram per ml concentration was used for subsequent analysis. For our UVA radiation model, A375 cells were exposed to varying intensities of UVA radiation, from 13 joules per centimeter square to 20 joules per centimeter square. After 24 hours of irradiation, the MTT assay was carried out to determine cell viability. The results show that UVA radiation decreased cell viability significantly, with values from 15.7% to 11.7%. Hence, the 13 joules per centimeter square dose was selected for further studies. At this dose, RS production was assessed using the DCFDA assay, while DNA damage was evaluated using DNA fragmentation assay. Simultaneously, EA treatment post-UVA irradiation was carried out for these two experimental designs. RS production was seen to increase 10.8-fold upon exposure to UVA radiation compared to the naive control, and with EA treatment at 40 microgram per ml, RS reduced to 5.44-fold, although this reduction was not significant when compared to the naive control. For the DNA damage, UVA radiation induced smearing in the A375 treated cells, indicating DNA degradation, and with EA treatment, this smearing was still observed, suggesting that EA did not confer any observable genoprotective effects in this condition. In conclusion, this study showed that UVA radiation induces cellular damage in A375 melanoma cells, and that EA treatment reduced RS production, but this reduction was not significant. The same can be inferred for the DNA damage assay, as smearing was observed in the EA treated cells. Future direction could explore antioxidant gene regulation such as catalase and superoxide dismutase. DNA damage could also be assessed more comprehensively using staining techniques like dye. Additionally, evaluating the protective effects of other antioxidants like vitamin C could provide further insights into the protective mechanisms against UVA-induced oxidative stress. Finally, I would like to thank Dr. Jessica Livingston, Mala McKenzie, Krista McFadden, Shanda Soli Chavez, and Dr. Michael Gibson for their guidance and support throughout this research. Thanks.